connection between a neuron and another cell that's being signaled. In the central nervous system, this is usually another neuron. In the peripheral nervous system, this can be muscle, smooth muscle, skeletal muscle, glandular tissue, or adipose tissue. And this can be named according to the type of tissue that's being signaled. So this can either be the neuromuscular junction if it's signaling skeletal muscle, or the neuroaffective junction if it's signaling the other types of tissues. Now, if one neuron is signaling another neuron, the first one we describe as the presynaptic, the second one is the postsynaptic, and we also have different types of connections. So we can describe an axodendritic connection. This is a connection or a synapse between the axon or the axon terminal of one neuron and the dendrites of a second or postsynaptic neuron. We can speak about an axosomatic type of communication between the axon terminal and the soma or the cell body of another neuron. And then we can speak about an axo-axonic, which is a very rare connection between both the axon terminals of two adjacent neurons. Okay? Now, most of the synapses that we're going to speak about are axodendritic. So between the axon terminal and the dendrites, the receptor cells on those dendrites, and they're usually unidirectional synaptic communication that we're going to be referring to as we go on. Now, synapses can be electrical or chemical, and we'll describe those in just a minute. But I want to kind of jump to this slide to describe these types of connections. So we talked about the axodendritic, which is this connection. So this is the most prevalent connection. This is what we're going to be referring to as we continue on to speak about synapses. This is between the axon terminal and the dendrites, the receptive dendrites of the neighboring neuron. So this is the presynaptic. This is the postsynaptic. We can also have two other types of connections, the axosomatic, between the cell body itself and the axon terminal of that other neuron. And then we can have a very rare type of connection called the axo-axonic, between both axon terminals of different neurons. Now, this axo-axonic synapse is usually involved in modulating neurons. And I'll talk more about that as we go on further. But this type of connection can be found as a regulating factor at other types of connections as well. We'll talk about modulating neurons. Okay. So in this image, we can see the main differences between electrical, which is shown up here, and chemical, which is shown down here, these two types of synapses. And as we can see, electrical synapses happen between the cytoplasm of both cells. There are these connecting structures called gap junctions, which facilitate bidirectional movement of electrical activity, whereas in the chemical synapse, we don't have that gap junction. We have the axon terminal and the neighboring cell, and we have exocytosis and chemicals being released at this junction. And so that's what differentiates an electrical synapse from a chemical synapse, is the presence of chemicals. So we're going to speak about some chemicals that are present at chemical synapses. They're called neurotransmitters, and they're usually present here, but not here in electrical synapses. So electrical synapses, this is all we'll say about them. They are usually linked by gap junctions, one neuron to the other. They usually facilitate rapid communication, just because we don't have to change the form of energy here. So we're transmitting electrical energy, and it's going to maintain its form as electrical energy. And so we don't have any chemicals involved here. So this is a much faster or rapid mode of communication. We use ions as second messengers and not chemicals. This is bidirectional, so this can happen going from the presynaptic to the postsynaptic back to the presynaptic in either direction. This can be an excitatory message or an excitatory communication, or an inhibitory communication at the same synapse. And then we can identify some examples of where this is found. So the retina is one example, the cortex, parts of the brainstem, the hypothalamus, where we have those neuroendocrine communications. Now, a whole lot is not understood about electrical synapses. At first, early physiologists were not sure that they existed. And now all that we know about them is that they're located in these few areas of the body. And then they're also found to some extent in the developing brain. So the developing brain of a fetus does show more extensive electrical synapses that are then converted to chemical synapses as that fetus develops. Okay, chemical synapses, on the other hand, are slower messages or slower modes of communication. We have different variations, as I mentioned earlier, so the axosomatic, axoaxonic, axodendritic, and we're involving two neurons which are speaking to each other, and they're using chemicals to relay these messages. So not ions, but chemicals to relay these messages. And so the electrical energy that is being passed down the length of the axon has to be converted to chemical energy when it gets to the axon terminal. And that's what kind of delays this process and makes it a bit slower than electrical synapses. So the synaptic cleft, which is the difference or the distance rather between the axon terminal and the adjacent structure, in this case, the dendrite, is usually about 30 to 50 nanometers wide, so a very small distance for those chemicals to move across and signal to the neighboring cell. And as we talked about before, this is going to be unidirectional communication because we cannot have action potentials traveling in the reverse direction. We cannot have chemicals traveling in the reverse direction. And the most common type of communication we'll speak about is the axodendritic communication, okay? Okay, so we're going to move into the chemical synapse in more detail. We'll speak about the anatomy first. So let's kind of remind ourselves of the big picture here. So this is our first neuron, the presynaptic neuron. This is that second neuron, the postsynaptic neuron. And if we zoom in really closely at this synaptic cleft, we can see what's happening on either side of the cleft here. So this is the cleft, the synaptic cleft, the space between that connection. And there are a few important structures that are present that are going to be involved in the synaptic communication. And we'll talk about those structures first, and then we'll kind of illustrate how they play together, how they work together. So we've got the neurotransmitter. Now the neurotransmitter is located inside this synaptic vesicle. And that is where it is stored until it's time to be released. And we can have different types of neurotransmitters that signal different types of messages, but the most abundant one that we will speak about is acetylcholine. We can also have epinephrine as a neurotransmitter as well. We'll talk about those molecules as we go on. We also have voltage-gated calcium channels. When we spoke about all the different types of channels along the neuron, we spoke about these voltage-gated calcium channels, and we mentioned that they were only found at the axon terminal, and this is where they are involved in this synaptic communication. We've got receptors on the adjacent cell, so on that postsynaptic cell, we've got receptors here lining this membrane. These can be of different types, but again, they're going to be specific to whatever this neurotransmitter is. So if this neurotransmitter is acetylcholine, this will be a cholinergic receptor to specifically bind that neurotransmitter. And then we have enzymes that are present here as well. And enzymes are basically involved in degrading any excess neurotransmitter at the cleft. We'll talk about the role of these enzymes in ceasing this communication. And lastly, we have reuptake molecules here, and they're usually present on the presynaptic side. And the reuptake molecules, as their name suggests, are involved in reabsorbing or uptaking excess neurotransmitter, again, as a part of ceasing this communication, of ceasing this signal. Okay. All right, so let's draw that out. Actually, let's walk through these steps here to wrap up, and then next time we meet, we'll kind of illustrate that a little bit better. Okay, so we've got the action potential coming down the length of this neuron. And when it gets to the axon terminal, it's going to trigger the opening up of these voltage-gated calcium channels. And that allows calcium to rush into the cell. and it depolarizes the membrane, which facilitates the exocytosis of these vesicles. So the vesicles move closer to the membrane, they merge with the membrane, and then they release their contents out into the synaptic cleft. After this neurotransmitter is dumped into the cleft, it has the opportunity to cross the cleft and then specifically bind to the receptor on the adjacent neuron. Now, the binding of this receptor with this neurotransmitter is going to bring about some type of response on this cell. We're going to speak in more detail about what that response is and about whether it's a slow response or a fast response. Also, the response can be a positive or a negative change in that adjacent neuron. Let's speak about that going on. Now, in order to cease this signal or stop this communication, we have two important structures that are involved. One is going to be this enzyme. So the enzyme is going to degrade or chop up any excess neurotransmitter into smaller pieces that can either be diffused away from the cleft, or they can be reuptaken into the previous neuron, the presynaptic neuron, via this reuptake molecule, where they can be recycled and repackaged to make some new neurotransmitter vesicles that can then be used again. Okay, so this is the sequence of events that happens where one neuron signals or communicates to the other neuron, transferring that electrical energy, which is the action potential, to chemical energy, which is the exocytosis of that neurotransmitter. And then that can then be reconverted to electrical energy on the second neuron down